Hi, I wanted to take a little time today to talk about that LD50 lab that we did before we went on this hiatus. We started preparing the Petri dishes with the seeds, and then we were supposed to come back on Monday, March 9th or whatever, and record our data, and then you were supposed to do the lab after that. So I went in and took pictures of all the data to give you the opportunity to maybe make something useful out of that. And the pictures that I took didn't include a really good representative sample, so I went back in later and checked everything out again and dealt with all of those Petri dishes, which I'm glad that I did because some of them were a little moldy, so I'm glad I got that taken care of. But I wanted to talk through some of the things about the LD50 lab that we didn't really get a chance to discuss in class. So first of all, just to review, LD50, LD stands for lethal dose, and 50 is 50%. So it answers the question, how much of a particular toxin do you have to have before it's really, really toxic? What would kill the median average organism? So sometimes we call it median lethal dose. Sometimes you'll see it written as LC50, lethal concentration for 50%. It's the same thing in any case. So, and I'm going to post this PowerPoint so you can look at it if you'd like to later as well. So we did a bioassay. Bio means living thing, but an assay is like a test to determine how much dosage you need to have a toxic effect. And so what it did is to help us produce a dose response curve. I think it's pretty likely that you might see a dose response curve on the AP exam and be asked to interpret that data. So we did we were looking for toxicity of salt on, this said radish seeds in the original lab, but we actually did alfalfa and lettuce, I believe. One of the two, the seeds were really old and they didn't give us very good results at all. They had really low germination rate. So these are some of the pictures that I took for you right when we first stopped going to school to give you an idea of what you're looking at. So here's a low concentration. You can see most of the seeds did germinate, and most of the radicals, the first bits of root that come out, most of the radicals are very long here, probably, 6 centimeters or so. At a 2% concentration, we had fewer that germinated and also shorter radical length. And then by the time you get to 3% concentration, you can see nothing germinated there. So this is what our data looks like after we put it all together. My son and I went back in and right after spring break and looked at all the seeds. Actually, it was right before spring break. We looked at all the Petri dishes and we took them apart and threw away all the nasty stuff. But we counted all the seeds that had germinated in every single set of Petri dishes. We did not measure every single radical to see the length. So we don't have that data here, and I think that's okay. So you can see that our concentrations here are increasing. Remember, when you're writing about experimental design, having that increasing concentration is really very helpful. Okay, so we've got the data here. You can see that we have increasing concentrations from our control, which is what we're going to compare everything else to. So our control in this case was a Petri dish with 10 seeds in it with 0% salt concentration. So we just used distilled water for that. And I found 7 Petri dishes that had some good, useful data for me. And in the 7 Petri dishes that were controls, each one should have started out with 10 seeds, and I'm assuming that that's the case. Of those 70 seeds, 12 of them did not germinate, and 12 out of 70 means that's a 17% mortality rate. So what that tells us is that on average, these seeds, not every single seed is going to actually germinate. And so we should assume that 17% probably won't germinate. So that's why you need a control, because if you looked at 0.01% concentration here, and over here we've got that 18% mortality, it almost looks like this salt killed off 18% of the seeds. But actually, when you look at the control, 17% is pretty normal. So is the salt really affecting the seeds at this 0.01% concentration? I would say if it is, it's probably not much. And if we had a much larger sample size, we might even find that that amount of salt doesn't have any impact at all. So then we have increasing concentrations, and you probably should expect that we'd have increasing mortality. So the next part of the lab that I asked you to do is to make a graph. And you could do it a few ways. If you used Excel, it might look something like this. So I'm going to use Excel to graph the data that I just showed you. Actually, I've already done it, so here we go. So I put in what I want on the x-axis here, that is the concentrations, and then on the y-axis is going to be the percent mortality. Why do we know that the x-axis is going to be the concentrations? Because that's our independent variable. That's the manipulated variable. That's what we as the researchers changed from 0% concentration increasing up to 3% concentration of salt in the water that we gave to these seeds. What depended on that, what's the dependent variable, what is the responding variable, is the mortality of the seeds. So the y-axis needs to be the mortality rate. So I've got this data in here with the x-axis first and then the y-axis, because that's the way Excel prefers to have the data, and that's usually the way you write it anyway. Here we have that same data, and I asked it to give me a scatter plot of it, and this is what we would call a linear graph. It's on a linear scale. This is the kind of graph that most people would draw if you were asked to draw a graph of this. So we have from on the y-axis from 0 up to 100% is where it should stop. Excel doesn't realize that we're talking about percentages here. So obviously you couldn't have mortality of 120% of the seeds, but that's okay. And then we have even spacing along the x-axis, making this a linear graph. And I'm going to pause there for a second. Okay, so we've talked about how to make a linear graph, but actually sometimes it makes more sense to look at this data using a logarithmic graph. Logarithmic graph is a little bit different. I'm going to see if I can move this. Let's see if I can put me over there. Okay, I think that worked. We'll see. So if you have a logarithmic graph, what you're ending up doing is taking, actually this is a semi-log graph, because we only did logarithms with the x-axis here. So we spread out the data to see if we can see a little bit more detail in this sort of middle section, because that's really where the important bit of information that we're trying to find is going to be located. So instead of going 1, 2, 3, 4 along our x-axis, or I suppose 0, 1, 2, 3, right? We're doing powers of 10. So 0.01, 0.1, 1, and 10. And then you see these little hash marks at the bottom, and those are the numbers that go in between. So let's look at between 1 and 10 here. So they're spaced a little differently because, again, it's powers of 10. So here's 1. This line is 2. This is 3, 4, 5, 6, 7, 8, 9. They get closer together, and then you get 10. It is possible that you'd be asked to draw a semi-log graph. I'd be really surprised to see that you would have to draw one. You should be able to interpret it, though. Okay, so in Excel, you make your graph, and in either case, you can add a trend line. So I added a trend line here, and you can kind of see in this one you get something of a J-shaped curve. But if we were to have continued our data out, it would go back into, it goes from exponential to a logistic curve. It's not going to go above 100%, of course, because you can't have more than 100% of your seeds not germinate. So, so we've got our trend line, and that's probably the best thing that you'd want to use to answer the question, what is the LD50 of salt for this particular type of seed? So let's see. I added in these lines here so you can see a little better. And what did I do here? Oh, I gave these guys some X and Y axis labels because, of course, if you're asked to draw a graph, you need to make sure you label everything. Got a nice title here that describes, LD50 graph. LD50 is what we're trying to find. And so then you usually write for your title whatever your x-axis title is versus your y-axis label. So concentration versus percent of seeds not germinated or something like that. So here's the big important part. We were asked to find the LD50 for salt. LD50 means it's the dose that kills 50%. And actually, if you go to the 50% mark, which is halfway between 40 and 60 here, and you make this line across, you will find no points. None of our points, none of our data actually is going to provide us the answer. But we can tell, here's some data here, we had a 1 and we had a 1.5, I guess, was somewhere over here. And right, this is 2 and 3, so this would be about 1.5. So we had some data points that are kind of close to our LD50, but they're not exactly on there, which means we're going to have to interpolate between those points and figure out where that is. Well, now it gets a little bit tricky. Should we use the trend line and say the LD50 is here, come down to here to report our data? Or should we use the dot-to-dot line that actually connects our 2 points and come down and report our data here? Well, the good news is with graph interpretation, they're going to allow some variation, and they would accept a range of answers, and either of these points would fall within that range. The question is really, do you understand that you go to the 50% mark and you go across to where the lines meet, and then you come down here? So hopefully, yes, you understand that. But let's discuss just a second here what these 2 lines would be reported as. So let's do this first line first. This is the one that corresponds to our trend line. So it's probably, I would say that would be the better answer. So if this is 0.1, our log, semi-log paper here, 0.1, so here we have, we're trying to find what value we should record for this red line here. So we have 0.1, this is 0.2, 0.3, 0.4, 0.5, 0.6, 0.7, and this falls right on the 0.8 line. So 0.8, remember these concentrations are in percents. So it's between 0.8 and 1 point, well, this is 2 and this is 1, so it's going to be something small. So I would say anything between 0.6 or 7 and 1.5 or so would be an acceptable, within the acceptable range for this question. So now what would you do if you were not real comfortable with that log graph? Or what if they didn't ask you to draw a log graph? What if they asked you to just draw a linear graph? And so this is going back to the original graph that we had. You can still interpret LD50 on this one. It might not be as precise, but it's still useful here. So again, you're going to find the 50%. I've lost my axis labels. That's okay. 50%'s here. So 50% mortality, go across, and then you come down, and it's going to be somewhere between 1 and 1.5. And again, that's going to fall within the range of what we had before. One other thing that they might ask you about on an LD50 or a dose response curve graph, and I created some fake data to get a better answer on this one, is there a threshold, and what is the threshold, if there is? The word threshold in the case of a dose response curve means the concentration where you first start to see some effect. So if you look at the data again, let's go back to our data from our original stuff, you can see that when there's zero concentration, 17% of the seeds failed to germinate. So when do you start to see that number come up? And looking at our data, it's somewhere between 0 and 0.05, because there's a big jump here between 18 and 27% mortality. Maybe there's a jump here, but it's kind of hard to say because it's only one seed difference. So this may be due to random chance. This jump maybe is not. But if you're looking at some graph data, you want to ask yourself when you start to see the curve moving up. And so looking at the data that I put in for this graph, you start to see a jump right about here, especially when you've got this trend line and you can see that all of these points are slightly below the trend line. Suddenly you've got one quite a bit above. So I would say that the threshold is somewhere between 0.01 and 0.1. So again, we're on a log scale here. So 0.01, this might be like 0.02, 0.03, something like that. And again, there's a range of answers that they would accept. So the term threshold of toxicity is when do you start to see, what is the lowest concentration where you start to see some kind of change, some kind of impact. LD50 again is the lethal dose for 50% of organisms that we tested it on. And then how does salt concentration influence the vigor of seed germination? The higher the salt concentration, the lower the germination rate. So make sure you answer that question by saying, as this one goes up, this one goes down. Or as this goes up, that goes up, or whatever. Talk about the relationship in a directional manner. You can't just say salt concentration determines how much the seeds germinate, right? You got to say more salt concentration, less germination. In this case, it's an inverse proportionality. Then there was a question that says, discuss the statement, dose makes the poison. So how does the dose make the poison? Well, keep in mind, anything can be toxic, even water. But you got to drink a huge amount of water in order for it to, well, the result is that it causes your blood to be too thin, and the concentration of all the other things that should be in your blood that aren't water to go down so far that you die from it. And I'm out of time on this one. Okay, so anything can be toxic. Water has a toxicity in LD50 of 90,000 milligrams per kilogram. So if you figured out your mass in kilograms, take your pounds divided by 2.2, and then multiply that by 90,000, that would be how many milligrams of water you would have to have before it could potentially be deadly. And that means that it would kill half the population. I haven't done the math today, but it's a huge amount of water. And so you're probably not going to die from drinking too much water, right? Sucrose has a smaller toxicity, but 29,700 milligrams per kilogram is a huge amount of sugar, not as much as the amount of water, right? It's more toxic than water, but it's not very toxic. These tell you, by the way, I got all this data from Wikipedia. My bird clock is singing to us right now. And this tells you the animal and then how it was introduced. And I don't know for gasoline if they did oral route for this. I would assume so for this rat test. But hey, you shouldn't drink gasoline, but actually it's not incredibly toxic compared to other things. Even vitamin C, which is, of course, good for you. Too much of it can be a problem. Glyphosate is Roundup, the herbicide, and it has a fairly low toxicity. There's concerns about long-term exposure, that chronic effect versus acute effect. But acute exposure for glyphosate It's a really fairly safe substance. I just pulled a few of these off of the Wikipedia site. If you open up the PowerPoint, you can check them all out if you're curious. Nicotine is, compared to water, obviously it's way more deadly. 3.3 milligrams per kilogram here. And the Shiga toxin, which is what makes you sick if you have dysentery, 2 nanograms. That's 2 times 10 to the negative 9 grams per kilogram. It's a tiny, tiny amount that's required to kill people, and that's why dysentery is such a big problem. So 2 environmental impacts of sodium chloride was the last question on your lab, and I thought I'd take a minute to discuss that. So obviously sodium chloride, the point we learned here is that even fairly low concentration, 1.5 or so percent of salt in water is going to be enough to stop seeds from germinating. And in coastal aquifers, as the sea level rises, salt water tends to get into the aquifers, and it's making the aquifers, which are currently full of fresh water, increasingly salty. If you're using that aquifer water near the coast as irrigation water, then you're going to have a couple of issues. First of all, you're going to have the salt water directly maybe causing seeds to germinate less. So if you're a farmer doing irrigation, that's going to be a problem. Second of all, as you know, if you irrigate a lot with water that has minerals in it, those minerals will build up. Some of the water will soak in, some of the water will evaporate, but the minerals kind of stay at the surface. And so that's called soil salinization. And that's a huge problem, huge concern for farmers as well. The other concern is, as desertification happens, areas that were maybe grasslands are becoming drier because of climate change. These places are going to need more irrigation water. And again, if they're using irrigation water that has any minerals dissolved in it, salt or anything else, they could end up with having difficulty getting their seeds to germinate. And I think that's all I got. So if you have any questions about the LD50 lab or soil salinization or how saltwater affects seeds, let me know and we'll talk in a Zoom sometime soon. See you later.